Good afternoon, everyone. We are going to be talking about DNA replication and maybe repair. We'll probably not get to that part today. So just a warning, today you may need a sheet of paper or two. We're going to draw a big picture of the DNA replication machinery. So make sure you have a piece of paper for that and you'll be in good shape. So let's talk about DNA replication and repair. So some of these things we've kind of hinted around in the lab, and so we're going to get into this in a lot of detail here. And maybe just before I move on, I'll mention kind of what the plan is for the next couple of weeks of the semester. Obviously, next week, Wednesday and Friday are the breaks. We have Remembrance Day on Wednesday, and Friday is one of the reading days, but we will have a lecture on Monday. And we have basically 20 units in this course. So it seems like a lot of material. In some ways it is. We have a lot of technical material with the DNA replication and some really interesting stuff. The last two topics are on biotechnology and viruses. So we'll get a chance to talk about COVID-19 a little bit. Most of these lectures are one topic. Some of them are relatively light. A couple are relatively technical. But like I had said before, there are three things you must learn and learn them well. That is DNA replication, transcription, and translation. So I'm going to emphasize that again and again and again. So hopefully that gives you an idea of what to expect. The other thing to expect is that I'm going to be giving you lots of hints, and some of them will be promises about the final exam. So I suggest that as we go through the next couple of weeks, maybe you have an extra piece of paper, and every time I throw a hint down, you write that down, because you know, when I make hints or promises, I do put things on the final exam, and some of them are going to be very straightforward. So hopefully you'll have, there'll be no surprises on the final exam is my goal, that everyone will know exactly what to expect. The other note before I continue is I just wanted to mention the midterm. I have almost finished grading them. I was just working on it now. I'm willing to bet I could probably be done them in about half an hour to 45 minutes. I just got a little bit behind schedule. And I realized some people are concerned about the grades. And I know the drop date, I think it might actually be Monday. So I will make sure I have the grades posted tonight. So my plan is after this class to just kind of work on them, and hopefully I'll be done by 6 o'clock. And have the grades up. So hopefully that will help you make any decisions. If you have any questions about that, whether you're not sure if you should drop or not, get a hold of me and I'll tell you my opinion on that, and if that can help you. Okay, so anyway, so sorry I didn't have those done. Like I said, my goal was to have them done, and I just got a little bit behind schedule with other things, but I guess it's just everybody this time of year. Everything has to be a little bit more patient because everything seems to be a little bit different this year. So let's talk about DNA replication. So we kind of finished off last day talking about Watson and Crick and how Watson and Crick made this observation that, hey, you know, if you have two strands of DNA, you just need to know what one strand is to figure out the other strand. And this is a possible replication mechanism. So they proposed all you need to do is separate the strands and put in the new nucleotides. And we talked about another experiment by, what is it, Nielsen and Mendelsohn? I can't remember the names. And we mentioned that they had proved that this indeed was the way that it was replicated. But of course, that didn't elucidate the entire mechanism. So that's what we want to talk about today, is the mechanism. So first part to consider about DNA structure is, what does it mean that DNA strands are complementary and anti-parallel? So those are some important concepts. So before we go on, I just want to review those two things. So complementary. What does complementary mean? It means that we have our base pairing. So of course that means we have A pairing with T and G pairing with C. So adenine and thymine have two hydrogen bonds, as are seen on that diagram. So the hydrogen bonds, remember, are these little dashed lines, and G and C has three hydrogen bonds. And so that's what complementary means. It means that the strands are not identical, but they are complementary. So complementary base pairing. And what was that 3' to 5' and all that stuff? So anti-parallel means that one of the strands runs in the opposite direction to one another in terms of the chemistry. So remember that 5' and 3' I talked about last day. We're talking about the carbons on the deoxyriboses, carbons on the sugars, and we're just numbering the carbons. And that's what organic chemists like to do. They like to number the carbons. And so the one strand, carbon number 3, starts at the top, and carbon number 5 is on the bottom. And then the other strand, carbon number 5 is on the top, and carbon number 3 is on the bottom. So that 3' to 5' stuff, and 5' to 3', we're going to talk about that a lot. So in fact, this is the one thing to know, is that everything in molecular biology goes 5' to 3'. It's kind of like the English language. We read our words from left to right. In DNA language and RNA language, we read everything from 5' to 3'. So you can see there's a note here. It says the enzymes can only add nucleotides to the 3' hydroxyl group. So I got a slide about that. And that should make sense. Enzymes have very specific substrates, so they can only bind one thing. And so like I said, I want to talk about that in terms of the enzymes. There's kind of a zoom in just showing the carbons labeled. I guess I forgot I had that. So 5' to 3' and 5' to 3' there, but anti-parallel to one another. So let's talk about those enzymes, and I have some notes I want to make about DNA polymerase, so you might need a piece of paper for that in a moment here. So it says here, nucleotides are added to the chain in the 5' to 3' direction. So there is a chain, and like I said, this little 3' OH, that 3' alcohol group, is the substrate for DNA polymerase. The other substrate for DNA polymerase is this thing here. So this thing here I'm going to call a DNTP. I'm writing up there because there's space. I'll put an arrow. So what is a DNTP? A DNTP is a deoxy nucleotide triphosphate. So for example, we could have DATP. So normal ATP is not a deoxy. It has the extra oxygen on it. And we could have DTTP, DCTP, and DGTP. So there's 4 of them, right, for the genetic code, the A, T, C, and G. So these are 3 phosphates. So the enzyme is going to grab this DNTP. It also grabs that 3' hydroxyl group. It hydrolyzes the triphosphate, and so you can see a diphosphate is released. And now we have a new bond. So this is our phosphodiester backbone on DNA. So like I said, I want to make a couple of notes about DNA polymerase. These are some important things to think about when we're talking about DNA here. And the enzyme here. So I have a Word document I was going to start. Hopefully that's shared with you. And I have some notes about DNA polymerase here to say. So I'm going to make them bullet points. I think I've got about 4 or 5 points to make about this. So DNA polymerase, okay, so we can say, don't want that underlined. There we go. Has 2 substrates, okay. And so what did I say those substrates were? The first substrate is, of course, the 3' OH. 3' OH end on the growing DNA strand. Okay. And the other substrate are the DNTPs. And you can list them again if you want. So we've got DATP, DTTP, DGTP, and DCTP. Okay, so that's the first thing to note about DNA polymerase. Something else to know is, okay, kind of what it does in general, maybe that should have been my first note, is it basically it adds complementary nucleotides. So maybe it's worth underlining complementary, right? Adds complementary nucleotides to the 3' end of the growing strand. Okay, so remember, it's got to be complementary. We're not just adding in any old nucleotide. They have to look at a template strand and see, okay, there's an A over there, so I'm going to add a T now. There's a G over there, I'm going to add a C now, and so on. Okay, so like I said, there are a couple other notes about this, is that this thing here, it requires a primer. So what is a primer? So a primer is a free, maybe free is the wrong word for this. Somebody says they can't hear me. Hopefully everyone else can. So unfortunately, if that's not true, I think hopefully everyone else can hear me. If it's your Wi-Fi, you may have to tune into the recording here later. So it requires a primer. So a primer is a free 3' OH end to get started. So it's kind of like a train has to be on the track to get going. In this case here, the DNA polymerase can't just start making DNA out of nothing. It has to have an end to grab onto. And so that concept's going to come back in a few minutes on that. Something else about this is it can proofread. So that's a good thing. We want our DNA to be copied, and we want it to be copied well without errors, so it can actually proofread. And what else? I thought I had one more note about this. Let me just check my notes here. Yeah, so in the last note I have on this, and like I said, we'll come back to these concepts in the lecture, is that this thing here requires the support of other enzymes and proteins. And so we're going to talk about all of those enzymes and proteins. We have 6 enzymes and proteins that we need to know a little bit about in total for DNA replication. So I'm going to switch back to the PowerPoint here in a moment. As always, if you miss this, you can tune into the video recording, or you can always request it from me. I can email you any of these notes if anybody wants. It's just complicated if I have to post them all the time. But like I said, you can always request any of these at any time. Okay, so back to the PowerPoint. Here we are, all my notes on this PowerPoint, and that's the job of the DNA polymerase. So it's really the most important enzyme here because it's doing the most important job, which is the actual copying of the DNA. Or sorry, not the copying, but making the complementary strand. And so we're going to see how that's done today. So there's a little cartoon for you, and you can see it says here, no one envied the job of the DNA polymerase shift manager. And he says, hey, Jerry, can you take a second shift today? And he says, nope, boss, sorry, I can only do the 5' to 3'. So cute little cartoon for you, and now you know what that means. Okay, so let's zoom in and take a look at some of the details here. So the area where DNA begins is called where replication begins is called a DNA replication fork. So you can see we have one strand on the top and it's dividing, and we're making 2 strands underneath, and you can see that region of replication that's going on. So we're going to kind of zoom in on the DNA replication fork as we look at what those enzymes are doing. So let's just take a look at some of the differences between bacteria and eukaryotes. So here is here's a final exam hint. We're talking about bacteria and eukaryotes a lot. And we have been way back, we started back in topic 5 and we keep going on. And so there's definitely, I usually find some sort of question on the final exam that's asking you about these things, some of the differences. So take some notes of that as you go along. So if you take a look, there's something here I want to point out. There's something here called an origin of replication. So what is that? That is actually a sequence of DNA, and that is where replication starts. So without that sequence, the DNA polymerase enzymes can't really bind and do their stuff. So you need that origin of replication. So that's where the replication is going to occur. And oh, there's a definition of it. And here goes. So it's going to divide, kind of split, and make 2 forks. You can see we've got a fork here on the left where replication is occurring. It's kind of going in that direction. A fork on the right, which is going in that direction. So it's going to keep continuing. There's our parent strand, the dark blue. The daughter strand is the lighter blue. And there it goes. little bit more replication, and it's going to go all the way around the entire chromosome. So in this case here, we have a circular chromosome. So eventually, there's 2 replication forks, and here we go. Eventually, it's going to meet up, and you're going to end up with 2 daughter DNA molecules. So there's an electron micrograph right there of the replication forks, or the replication bubble in the bacteria. So that takes, in E. coli, about 30 minutes, which is like really amazingly fast. Again, not as much, half as much DNA as we have. So eukaryotes, a little bit more complex, but before we get to eukaryotes, I just wanted to mention we have talked about plasmids in this class. So plasmids you can think of as mini chromosomes, and plasmids have to have their own origin replication as well in order to be copied. This is kind of an important concept for biotechnology, because if we want to copy DNA in E. coli, we can do this. It's actually relatively easily done. You can take an E. coli origin replication, you can add some artificial DNA to it. So you can see there's a circular DNA molecule, and now we can replicate that DNA in E. coli. And so the plasmid that we had in our class is an artificial plasmid with an E. coli origin replication, and it can be used for various biotechnology uses. So we'll talk a little bit about biotechnology now in this unit, but a lot more so in topic 19. So there's a plasmid. Something else we learned about in the lab is that plasmids are generally supercoiled. So you can see there's a micrograph of a supercoiled plasmid, which kind of just means it's twisted up and packaged up a little bit more efficiently rather than being a giant ring. So in eukaryotes, a little bit different. Our chromosomes are massive, and so we actually have multiple origins of replication in our chromosomes. And so you can see that's labeled there. We also have linear chromosomes, so it's not going to go around and meet itself. You can see we've got a whole bunch of, there's the parent strand, the daughter strand, and a bunch of replication bubbles. And there it goes. Eventually, the replication bubbles are going to meet one another, and you'll end up with your daughter strands, and there we go. So there's an electron micrograph of a bunch of replication bubbles in a eukaryotic organism. Okay, so, well, there's another electron micrograph for you. There's some very cool pictures. It's amazing we can do this. So there's our DNA polymerase. You can see we have our parent strand is the purple, and the new strand is in yellow. And I had some notes here about DNA polymerase, and I think these are probably similar, if not the same, to the notes I just wrote in the Word document a few minutes ago. So I mentioned it has 2 substrates. It requires a 3' end of the growing strand. And I guess that was the other thing I forgot to write, is that it does synthesize the new strand in the 5' to 3' direction. So you may wish to write that down as well. And I said this already, the new strand is made complementary to the template strand. So just some reminders, right? I'm writing things down. That means it's important. It's coming back. I'm saying it all over again. It means it's important. What else about DNA polymerase? It cannot start with a 3' end. So I mentioned that already. It requires a primer. It has proofreading activity. And I guess the other thing I didn't mention is that it can remove the primers as well. So part of that proofreading activity means it can remove things that shouldn't be there. So incorrect base pairs or RNA shouldn't be there. So it can remove the RNA in the end. And so you'll see that happening when we look at the process. So here's a diagram kind of similar to what I showed you before. The 3' end and the DNTP. These things are going to get together. Notice complementary T is matched with the A on the template strand. The DNTP is hydrolyzed. We end up with 2 phosphates being released. And then there's our final nucleotide added onto that growing strand. Okay, so let's zoom in on this DNA replication fork. So what I'm going to do is basically talk about what's going on here, and then I'm going to draw it for you. Okay, I know when people look at this initially, they're like, okay, what now is going on? And so it's kind of important to dissect this piece by piece. So hopefully by the time I'm drawing this thing, it'll make a little bit of sense, but I want to point out a couple of things in here. There are 2 strands. of DNA. So there's 2 replication events going on. This one here, of course, there's 4 replication events going on. You've got DNA being replicated into the right, DNA being replicated to the left. So it does look pretty complicated. So when we zoom in and make our replication fork, I'm just going to kind of do either the left or the right. I'll probably do the right. And you can see there's some other things. It's talking about the leading strand and the lagging strand. And I'll get to that in a moment. So key thing here is we've got 2 strands of DNA we're going to start off with, and they start at an origin of replication. Okay, so let's take a look at this kind of in some detail. Something else I'd mentioned is that there's a lot of supporting enzymes and proteins that are important, and all of these things will pop into the lecture notes here and there. So here's some of the supporting enzymes and proteins. So the first one to take note of is primase, and I'm gonna, when I make my drawing, I'm gonna make some notes on this as well. And I know there are definitions in the notes that I gave you as well. So first one is primase, and it is going to add a little sequence of RNA. So like I said, it just kind of, DNA polymerase, it can't start on its own. So it needs a primer. So it's kind of like, think about if you prime an engine, you know, sometimes people have a motorcycle or a snow machine or something like that, and there's a little primer that injects gas into the lines. Cars don't need that because it's done automatically by a fuel injector. So, you know, that's a little bit of automotive knowledge for you. But look at this, we've got a free 3' end. So RNA is kind of similar to DNA, so this works. It's kind of a temporary, just, hey, start here kind of thing. So primase does that. We also have helicase. So helicase is important, and it will actually unwind the DNA, right? It separates the 2 strands. So it should be called unhelicase because it's separating them, but I didn't name it. So it breaks hydrogen bonds. Once the hydrogen bonds are broken, we need to hold them broken apart until the DNA polymerase can get in there. So that's why we have these single-stranded binding proteins. And they're just kind of holding it single-stranded, kind of like placeholders. The last one here to take note of at the moment is topoisomerase. So what is that thing doing? It is kind of like an untangler. So you may know if you untangle something, particularly anybody who's had braided hair might be familiar with this kind of concept, is you don't just yank the whole thing apart. It's going to make knots. So DNA will do that as well. It's a helix. You try to yank it apart, it's going to form knots. So the topoisomerase actually kind of just unknots them. If you take a look here, you can actually see what it does is it makes a little nick right there and lets it untwist a little bit and then reseals the nick. So it's important for that, or else the DNA can't get unwound. So let's take a look at the process here. I'm going to start off and talk about the leading strand, and then I'll tell you what the lagging strand is here in a minute. So if you take a look, we've got a whole bunch of things going on, and let's just kind of take this apart piece by piece. So you have the parent strand. You can see we have the helicase right here. So there's helicase with an H, and it's separated the DNA. And you've got the single-stranded binding proteins here, so single-stranded binding proteins. And you've got this RNA primer that has been produced. And DNA polymerase, here it goes. It's going along. It's reading the template strand here. It's giving, it's putting in place complementary nucleotides. So you may notice this is DNA polymerase 3. There are a few of these. I am not going to expect you to know all the differences between the different DNA polymerases. That's not important for this class. So just call it DNA polymerase, but it does talk about them a little bit in the textbook. And so there you go. It's going along. It's traveling along the template strand, and it's filling in new nucleotides in the 5' to 3' direction. And so there's it going. It's elongated. And we'll talk about that other strand in a minute. And as it's going along, the helicase is kind of slipping along just ahead of it, and then kind of upstream is also that topoisomerase. So this is it, right? You got these 3 things to kind of get things going. You got the helicase, single-stranded binding proteins, and the primer, and then the DNA polymerase is free to kind of do its thing, go in and add new nucleotides. So here's just another diagram showing the same thing. Just wanted to show you kind of, this one's a little bit of the opposite direction, but we've got the primer starting there. And you've got the elongation going on. and the new nucleotides being put into place. Always in the 5' to 3' direction. And there it goes, keeps on going. And the last thing, the reason why I like this diagram in particular, was that you can see another DNA polymerase here comes along and is removing that primer, right? Because we're trying to make DNA. We don't want this RNA chunk here. So it's like, hey, let's get rid of that and we'll fill in some DNA. And then we have a complete daughter strand. So that's the leading strand. And so why is it called a leading strand? Because, well, it just kind of goes along and, you know, once it gets started, it goes until it gets to the very end of the chromosome. So the lagging strand is a little bit different. So we're just talking about this enzyme, and it only goes from the 5' to 3' direction. So this leads to some problems. Okay, so let's kind of zoom in here and take a look at what's going on. So if you take a look at this, we have a piece of RNA that's been replicated, and this whole thing here, it's separating over here in this direction. So if the enzyme can't, I'm going to draw something and then erase it. The enzyme can't zip along like this, right? It can't do that, right? That's a no. It can't go in the 3' to 5' direction. It can only go in the, can't erase this. I thought I could erase it with this pen here. Oh, there we go. It can only go in the 5' to 3' direction. So it kind of just does a little bit at a time, right? So you can see we have to make the RNA primer here. We're going to synthesize a little DNA, and then as it separates up over here where I put the asterisks, we're going to do another fragment. So it kind of just does it fragment by fragment, okay? So same steps, right? We have primase comes along and adds an RNA primer. The DNA polymerase adds the DNA nucleotides, and these little fragments that are formed are called Okazaki fragments. So they're named after a Japanese guy, so I'm assuming he was the one who discovered these or studied them. And so that's why it has the name Okazaki fragment. So there's one fragment. You can see the DNA polymerase pops off because, well, it ran into that RNA. And then here we go. You got another fragment is going to be made over here in this region, and it's going to need a primer, and it's then the DNA polymerase is going to go in and make another little fragment. And so now we have all these little fragments. So remember before I told you another DNA polymerase comes along and removes the RNA. So, so that's good because we want DNA. And then, so there it goes, replacing the RNA with DNA. Now there's a problem, right? When it finally gets to the end here, it cannot grab onto this end, right? That's not what DNA polymerase does. It can't just grab onto a substrate it doesn't recognize. So we're still going to end up with a tiny little bond missing between a 3' end and a 5' end. And so we actually have one more enzyme here that we have to deal with. It's called DNA ligase. So ligate in chemistry means joining two things together. This uses ATP, and it's going to seal those two fragments together using a covalent bond. Okay, so information overload, right? Hopefully you got some of it. I'm going to draw for you a DNA replication fork and kind of repeat some of that information. So hopefully you have a piece of paper, I think, we're going to draw some of it. It may look complicated, but not as difficult as you may think. So there's a diagram from the textbook. I'm going to kind of draw it a little bit different than what it's showing, but you can see it's saying, hey, don't forget there are 6 proteins and enzymes, and you are going to see these on the final exam. So I think last year I had a matching question. The year before I had some sort of fill in the blank question. There's always on every exam a 6-mark question about these. So make sure that you know what these things do, okay? Because I'm definitely going to ask you about them. So it says draw a replication fork on the board here. Okay, so we're going to do that. Usually I'm on the whiteboard, but I'll use the virtual whiteboard. So let's see here. I have it started. So, okay. So here goes. So what you can see, I've drawn 2 strands of DNA, and what I'm going to do is just zoom in here. So it says DNA replication fork. Just want to zoom in and add a couple of labels. And so all of my DNA strands are going to run in the 5' to 3' direction. So I have little arrows there. 5' to 3', and I'm just going to join these 2 together like that. Okay, so that's a good place to start. I have two strands of DNA. They're running anti-parallel to each other. One is 5' to 3', the other is 3' to 5', anti-parallel to one another. So that's good. And now just it's a matter of kind of trying to put in some of the details. So there's different ways to do this, because if you happen to draw this upside down, then you might have to do things slightly differently. But what I'm going to do is, like I said, put a lot of labels on this. And so this, of course, I'm going to draw something and then I'll erase it here for a second. So this DNA is, of course, going to be separated in that direction too, right? So this is a whole bubble. But what I'm going to do is, I can't erase that very easily. There we go. What I'm going to do is just draw one replication fork. Okay, so a couple of notes to put on this, right? So first thing is I'm going to put a little arrow here. And so what is my arrow all about? Just a reminder that this here is the direction of replication. Okay, so just keep in mind that that's going to separate out in that direction there. Okay, so next thing, I'm going to start at the very beginning, and the first thing we're going to do is we're going to draw helicase, because helicase is involved in separating those DNA strands. So I'll draw the helicase. Oops, what did I do? There we go. So sorry, I keep shrinking and growing things, but I'm going to draw the helicase, and it's going to go right here. How about? There we go. Okay, so that is helicase. So I kind of pre-wrote some notes for myself for this. So apologize, hopefully I'm not going to go too fast for everyone, but I'm going to kind of cut and paste some notes here. It's just I find it hard to write on the whiteboard here, but here goes. There's helicase, and there's my note about helicase. So I'm going to take that and just maybe shrink it just a little bit. So it says helicase separates the two strands of DNA by breaking hydrogen bonds. Okay, so there's my note for helicase. I'm just going to shrink that a tiny bit, make sure we can squeeze everything in. It's going to go just below helicase there. Okay, so there's the helicase. So the other thing, like I said, we've got those single-stranded binding protein things. So we're going to draw those as well. And let's see here what color we can use. I'll use kind of a pinkish color for those. So those single-stranded binding proteins are kind of just behind helicase on both strands. So there they are right there. I'm going to draw them like this. Usually I draw this massive. It's really big. So let me just zoom in a little bit here. Take up two whiteboards for this normally. Okay, so I'm going to put a note for those as well. So these are the single-stranded binding proteins. Okay, and I have a note for that as well. So my note says it binds single-stranded DNA and prevents double helix reforming. Okay, so here it goes. We'll pop that on there. I'm going to stick that above once I can figure out how to move it. There we go. I think I just had to click on one thing. There we go. Single-stranded binding proteins. Okay, so there's note number 2. Okay, so what else do we need to draw? We are going to draw the topoisomerase. I didn't leave a lot of space for it, but I will squeeze it in there somewhere. So the topoisomerase, remember, it is involved in making sure that it's not getting all tangled up. So there we go. I'm going to draw that one in purple, and maybe I will squeeze it in right here, just kind of upstream. So there it is. It's purple. Okay, and so I'm going to squeeze it in over here. Topo. Isomerase. All right, so I've got a note for that. It says it untangles and reduces the tension in the unwinding DNA strand. So attach that in there. There it is. And I can move that somehow. I need to figure out how. I just got to click on something else to move it. There we go. Topoisomerase. Entangles and reduces the tension in the unwinding DNA strand. Okay, so all those things are helping this thing to get going. And so let's get into the DNA replication. We want to add in our DNA polymerase, and we're going to start off over here. So I think my DNA polymerase, I'm going to do that in black. And we're going to start over here with the leading strand. Okay, so I'm going to zoom in. Hopefully you got the notes on the right there. I'll probably zoom out another point, but I just want to make sure I've got it looking really nice and neat. Okay, so my strand of DNA is going to get synthesized in the 5' to 3' direction. So I'm not going to talk about the primase quite yet. Okay, I'll get there in a moment. And so there's my double-stranded DNA that's getting formed. Okay, 5' to 3' direction, don't forget that. And my DNA polymerase, what I'm going to do is it's kind of just going to look like a black box like that. There's my prime. Okay, so this here is DNA polymerase. There we go. Okay, I've got to write a note for that. I know we wrote lots of notes for it already, but reinforcement is good. Remember what these things are, and I'm not going to write a huge note, just a little note on that. And there it is. So it says it adds new complementary nucleotides at the 3' end. So I will move that up top here in a moment. I just got to click around and move it there. There's my note for DNA polymerase. So that's, you know, pretty straightforward there for the leading strand. We kind of ignored our primase, which is going to which would have started things way back at the origin of replication. And maybe what I will do is I will squeeze the primase in here. So let me just move this over, and I'm going to squeeze the primase in over at the start there. So primase will be in red, and what I'm going to do is I'll zoom in again here. And my primase, here we go, it's in red. Remember, my little primer is also going to be in the 5' to 3' direction. Okay, and so maybe I'll just label that as the primer, and I'll come back to the primase in a minute. So probably it would probably be fine to squeeze it in right here. RNA primer. There we go. Okay, so there's the picture so far. Now what we have to do is think about what is going on with the lagging strand. And I'm going to kind of do the lagging strand a little bit backwards because I want to just think about in terms of the sequence of events that are going on. Okay, so the very first thing that's going to go on, so zooming in again, is the primer is going to be made. So I'll keep my red pen out, and there's my little primer. So my primer is, like all nucleotide sequences, is made in the 5' to 3' direction. Here we go. Okay, so I'll just put a little note that that is my RNA primer. Okay, and I guess I just need maybe a slightly different color for the primase. So what I'm going to do is choose kind of an orangey color here. So there's an orangey color. Oh, maybe it's not different enough, but that's okay. Hopefully you can see it just fine there. So this here, I'm going to zoom out a little bit so I can put in my note. Okay, this is my primase. There we go. Okay, so my note for primase, it says here, it adds a short segment of complementary RNA as a starting point for the DNA polymerase. So let's put that in here and squeeze it in. So shorten it. Okay, there's the primase note. So I think we're probably doing okay for space. Maybe I will shrink that just a little bit so it fits in there a little bit better. There we go. Okay, like I said, there's the picture so far. We got the primase. So the very first thing is that the primase is going to add that little RNA primer, right? So let's do another Okazaki fragment now. Okay, so we're going to go to the right, and so here is our next Okazaki fragment right there. So like I said, remember things are in the 5' to 3'. Now this particular Okazaki fragment, the DNA polymerase has come along and it's made a little bit of DNA. So there we go. So we're going to just have some RNA and we have some DNA there. And of course, this is DNA polymerase, so we can label that. DNA polymerase is going to be right here. I think I made it a square shape. Don't forget that's 5' to 3'. And I will label it up here. DNA polymerase. There we go. So we don't need another note for DNA polymerase, hopefully, because we made the note above. Maybe the one note we can make is that is an Okazaki fragment. So I just have to figure out exactly where I'm going to write that. Maybe I will write it, let's see here. I'll write it right here. We've got Okazaki fragment. There we go. So hopefully that is, the whole thing is an Okazaki fragment, the DNA and RNA attached together. Okay, so we're almost done. I'm going to draw one more fragment here at the end because, of course, we still have to talk about ligase, okay? So let's just zoom out or zoom in a little bit here. And so we have another strand here, 5' to 3', right? And then we've got another one here, 5' to 3'. So I'll write the little 3' down there and a 5' down there. Hopefully that looks like a 5 and a 3. It's kind of hard to squeeze things in there. Let's see here. Try that again with my 5. And this strand here, I can stretch out a little bit if I want. So I'll just do that so it's all in there. So hopefully you made yours nice and big, so we can squeeze lots of things in there. So of course we have our hydrogen bonds in there. And let's see here, I'll choose a new color. How about that color there? And it's going to be, I'll make it a square. How about, or sort of a, okay, that's a wonderful square. Hope you like my square. Okay, so that's ligase right there. So notice ligase is joining 2 DNA fragments because the DNA polymerase has removed the RNA fragment. So the last note here is from our ligase, and it just says it joins the 2 Okazaki fragments together. So you can mention that it's a covalent bond if you like. So there's my note. Just going to bring it over there. There we go. And there's my ligase. Okay, so there's the complete picture. Okay, so hopefully it wasn't too painful. You know, after going through these a couple of times, it does all start to make sense. So just wanted to make some last comments on here. I'm going to draw on this and with a highlighter to kind of just emphasize what's going on at the lagging strand. Oh, I got to put a note for lagging strand. So this is the lagging strand right here. There we go. Now our picture is complete. So just another note here about the lagging strand, right? So remember the origin and replication is kind of to the left of this. So that means that this strand here, oops, I did not mean to do that. And I don't know if there's an undo. There's an undo right there. There we go. So this strand here, this one that I'm highlighting in yellow, right? So that one would have been made first as the DNA started to separate. And then this one is going to be made, right? And then as it starts to open up, this one is going to be made. And then as this opens up more, we're going to have more strands, Okazaki fragments being made. So hopefully you enjoyed my artwork. And this is, like I said, this is something I usually do on the whiteboard and have, you know, takes up 2 full spaces, but sometimes it's worth it to draw these things out step by step to understand what's going on. In biology, there's a lot of complicated things, and if you draw things out step by step, it gives you an idea of what's going on. It's really important you understand what's going on with those 5' and 3' things as well. So I'm going to switch back to the PowerPoint. Hopefully everyone got all the important things there. And where's my PowerPoint? Oh, there it is. So it says draw a replication fork on the board here. And so here we go. This is from, I think, last fall. I drew this on the board. And so you can see it's not that much different. In fact, yours probably has a little bit more detail than this one. It looks like, but and slightly different color scheme, but basically all the same information there. Okay, so we're at 4:45, and that's incredible because it looks like last fall I was at exactly 4:45 as well. So yeah, it's nice to know you're consistent. So a couple other things about DNA replication. I have a video to show you, and I think I'm just going to save that for next day as a bit of a reminder how this happens. But here's kind of a diagram from the textbook that's showing that you know, DNA replication is even more complex than that, right? And when they're trying to show you kind of what the current model looks like and how the lagging strand and the leading strand are going together. And of course, there are some differences in eukaryotes and prokaryotes. I think in prokaryotes, the DNA polymerase is called DNA polymerase sigma instead of 3. So I don't know why the naming is different. But like I said, don't worry about the different DNA polymerases. That's not so important here today. So there's the video. Like I said, I'll share that with you next day as kind of a little review. And I think I just want to touch on DNA repair, and then I'll come back to some concepts in this unit here next day. So one thing to know is that the DNA polymerase has the proofreading ability, which is great because you know, it does make mistakes, about 1 in 10,000, which is pretty good. I'd like to see you copy something word for word and only make 1 mistake in 10,000. And then because of the proofreading and because of repair mechanisms, actually only about 1 mistake in a billion get through. So that's good, particularly, you know, if we have children, we don't want them to have a bunch of mutations, right, in their cells. Issues do happen. There are all sorts of reasons why DNA may need to be repaired. We get exposed to radiation, we get exposed to chemical carcinogens. There are all sorts of things happening all the time to our DNA. So it's important that we have a good repair mechanism. In fact, we have multiple repair mechanisms because our DNA is very, very important. There is at least 100 different repair enzymes and whatnot that have been discovered. I bet you that list is like 200 now or more because, like I said, we're trying to make sure we don't have any issues. So I just want to talk about repair kind of briefly here in sort of a general sense about what's going on. Just want to show you here, this is one type of damage. This is showing UV damage to thymines. And so if you take a look, here's thymine here, and here's a second thymine. And so sometimes with UV light, there's 2 thymines together. What it does is it rearranges those pi bonds and those double bonds, and they kind of attach together, and you get this little thing here called a thymine dimer. So that's a problem because your genetic code will not be read properly, and you're going to have a mutation in that gene. So we don't want that. We don't want cancer. We don't want bad genes. So we have a repair mechanism. And like I said, I just want to kind of generally explain to you how it works. So there's some damaged DNA. It looks like a thymine dimer. And we have enzymes that recognize the damaged DNA. So these are called endonucleases, meaning that they read sequences within the genome, right? So they basically nick the DNA. So that nick is kind of just cutting 1 covalent bond. And then everything else is really just our DNA polymerase and our DNA replication machinery. So DNA polymerase, it has the potential to remove RNA and bad DNA. Once it's nicked, it just needs to kind of get in there. It can add the new DNA in the 5' to 3' direction. And then eventually you're going to have a gap, just like the lagging strand, and ligase is going to get in there. So kind of the bottom line for DNA repair is that it's mostly using the DNA replication machinery with a little help from some endonucleases that are kind of just recognizing the damaged DNA and cutting some covalent bonds to help things out. Okay, so that is the end for today. So thanks for coming out on a Friday, and I know we're all looking forward to next week with a little bit of a break. So hopefully you get a chance to get caught up on things. And like I said, I'm going to actually try to get those midterms done right now. So hopefully you can get your grade. I say check back in about an hour, and hopefully I'll have those grades posted, and we'll discuss the midterm in more detail on in Monday's class. If you have concerns before Monday, like I said, I think Monday might be the drop day. Fire me an email, and I'll try to respond to it as soon as I can. So have a wonderful weekend. Enjoy the snow and November in general. See you later.